Okay. I'm Zang. I'm a PhD student from Leiden University, and I'm very happy to be here to share our current project, the age-related changes in neurovariability in a decision-making task. This is still a work in progress, and we're interested in the health aging process. This process is reflected at different levels. From psychological perspective, aging is associated with impairments in decision-making, memory, learning, and many other cognitive functions. Also, biological research found that aging causes cellular neurodegeneration, neural loss, and deficient neuromodulation, and so on. But how does biological change give rise to the psychological change? One potential way is those classical theories from last century. They state the neurovariability increases with age, and thus the effective signal-to-noise ratio in the central nervous system decreases with age, which may finally lead to the cognitive decline. However, those theories were not tested directly at that time. And recent research on anesthetized monkeys, they found, maybe I should use the point. Oh, sorry. A recent research on anesthetized monkeys, they found both the single neural variability and noise correlations increase with age in the cortex. Those results are consistent with the first part of those classical theories. However, they can't link the neural variability to cognitive decline because they are using anesthetized monkeys in their research. So in our project, we would like to quantify neural variability. We would like to explore neural variability in large-scale neural recordings from behaving mice. In this way, we can ultimately link the neural changes to behavior. Also, we want to quantify neural variability in other neural structures beyond the cortex, because those previous research, they were focusing on the cortex, and we don't know what's happening in other brain structures inside the brain. And our hypothesis is the older mice exhibit higher levels of single neural variability. Let's first look at the datasets we're using. We included the IBL brainwide map datasets in our current analysis. I guess you're already familiar with this public dataset in the past 3 days. And another part is a personal project, the Learning Lifespan project. But both datasets are recorded based on the same standardized protocols. And we then redivided those data into 2 age groups according to the age of mouse. And if you have no idea of the age of mouse, then according to the life phase equivalence, we found the human equivalence of those 2 age groups. The young group, the mean age of the young group is about 31 years old, and the old group is about 51 years old in human. And we first looked at the behavioral data. We first plotted the psychometric function for both age groups, and we found the older group showed slightly worse performance, which means they're less sensitive to the change of the different contrast level. And we then looked at the response time. This time we can see from the correlation here, not the median response time, but the response time variability increase with age, which means the older group isn't slower, they're just more variable in the response time. Here, we also plotted the distribution of response time in both groups. This result is consistent with previous findings in human. Here is a figure from previous literature. They found that across lifespan, the behavioral variability shows a U shape. But within this specific age range, this is the range covered in our dataset. We can see the behavioral variability slightly goes up. In another literature, we found similar pattern, but here the y-axis actually shows the 1 over behavioral variability. So here it goes down. but it contains the same meaning. And we then looked at the neural data. Here I plotted the trajectory of those probes in both datasets. And then we can look at here I indicated the regions we are interested in. Here actually you can see those 5 brain regions are from the repeated site, just as Martha just mentioned. And for the quality control process, we applied a set of quality control criterion for both datasets. Start from the session level, probe level, to trials level, and neural level. And we then divided those sessions with good quality into 2 age groups, as I mentioned earlier. But here is a very important reminder because in our current dataset, the different spike sorting versions are used, which means we have to be very cautious about our current result in terms of the neural data. Ultimately, we would like to run the same analysis based on the same version of spike sorted data. At this moment, we are more focusing on building up our analysis pipeline. And now we have the data. How can we really measure the neural variability? Here we summarize. Here is a summary of previous some widely used measures to quantify neural variability for different types of neural signals, from neural spiking data, EEG data to both signals. Specifically, for the neural spiking data, the final factor and the noise correlations are widely used. And we will start from the lowest level. We will use the final factor to capture the single neural variability. Here is the definition of final factor. It is defined as the spike count variance over trials divided by the spike count mean. So now we have the tools. I will use one brain region LP as an example to illustrate the data analysis we are using. And here is a LP brain region, the lateral-posterior nucleus of thalamus. And for each neuron and each stimulus level, we extracted all of those trials and then aligned it to the stimulus onset. Then, by using a sliding window method, we computed the average firing rate and the final fact time course for this neuron and this condition. We then combined all of those neurons and condition together and computed the group average for both groups. Then, based on those time course, we can make a comparison between groups. From the overall pattern, we can see the old group showed smaller firing rate, but a higher final factor in the post-stimulus period. And then we can see results from other brain regions. We can see in hippocampus and thalamus, the old group actually showed larger neural variability than the young group. However, in brain regions like PPC and ACA, the pattern actually flipped. And in some other brain regions like limbic and olfactory area, we didn't find group differences. But I have to mention, this is still very preliminary results, so maybe we shouldn't overinterpret them. And then let's summarize. In our current result, we indeed found the aging effects, and for the behavior, we found the old animals showed slightly worse performance and increased response time variability. And here is also a very simple summary, a summary of the neural variability. Yeah, but we have to wait for the final results. And in our next steps, we would like to explore the neural correlations between neurons. In this way, we can extend our current analysis from the single neuron level to the local circuit level. We're also interested in different cell types. ultimately, we would like to link those neural results to behavior to explore the behavioral consequences of neurovariability. And in our lab, some people are still also working on the human extension of the IBL task, which is also very exciting. Yeah, and in the end, I would like to thank our co-authors and our lab, and I'm very grateful for the IBL dataset I'm using. And thanks a lot for your attention.